Hi, you're again welcome to Muhumuza Naphtali, medicalsciences.com. And as I always encourage you, make sure you subscribe. Subscribe, you can like the video and comment where you need us to improve. And today we want to talk about introduction to enzymology. And this enzymology is covered both in clinical chemistry and biochemistry. So if you are a student of biochemistry, you are a student of clinical chemistry, you're privileged to have this video. Let us get started. So enzymology is the study of enzymes. We are looking at the study of enzymes. And these enzymes play a big role in our body. And you can ask yourself, what is an enzyme? And we know that the enzyme is a biological catalyst. Enzyme is a biological catalyst. It's a biological catalyst whereby this biological catalyst, meaning it is produced by the living cells, is a biological catalyst. And this biological catalyst is a protein in nature. Protein in nature. And the role of catalyst, it is always to speed up the rate of biochemical reactions. So these enzymes, their role is to speed up. They are responsible for speeding up the rate of biochemical reaction. The rate of biochemical reactions in living cells. In living cells. So we see these enzymes, they are found in living cells and they are biological catalysts. That's why we call them biological catalysts. And they are protein in nature. And they speed up the rate of biochemical reactions. And these enzymes have different properties. They have different properties. So we can look at properties of enzymes. Properties of enzymes. One of them is that they are protein in nature. Enzymes are protein in nature. Protein in nature. And these enzymes also, they are denatured or affected. They are affected by temperature. Temperature, they are affected by temperature. They are affected by pH. So you find enzymes work best in their specific pH and at optimum temperature. So these are enzymes. And another thing, these enzymes, they remain, after the end of the reaction, they remain the way they are, or they remain the same at the end. They are not used up in the reaction. They are not used up in the reaction. These are the enzymes. And these enzymes, they are produced by living cells. Produced by living cells, stroke tissues. So these are some of the properties of these enzymes, which we normally determine in the laboratory. We have seen drugs being used containing enzymes, and we are using them in diagnosis of diseases. And they are solely produced by the living cells. So we shall see enzymes are found in tissues, not in blood circulation, but they are found in high concentration in cells or tissues where they are produced. They only become much in blood when there is tissue damage or cell damage. And we want to see the mechanism. How do these enzymes work to speed up the rate of biochemical reaction? And we want to see the mechanism of action. Mechanism of action of enzymes. Mechanism of action of enzymes. And these enzymes, they work under lock and key. Remember, Even in secondary, they taught us that they work under rock and key hypothesis, rock and key, whereby if I have the rock, then the key should be complementary to this rock so that it can open it. So we see if we have the enzyme, if this enzyme is acting as a rock, this is the enzyme acting as a rock, then the substrate should be complementary. The substrate should be complementary, or it should fit in the active site of the enzyme. Then when it can bind to the active site to form an enzyme, substrate complex. Enzyme substrate complex. We form this enzyme substrate complex, and after forming the enzyme substrate complex, at the end of the reaction, we form a product. We form product and we release the enzyme. The enzyme is unused. The enzyme is released to go back and catalyze other reactions. Then we form so the major work of this catalysis is that we are going to form a product. And how does it speed up this rate of biochemical reaction? By this lock and key hypothesis, is that it speeds up the rate of biochemical reaction by lowering activation energy. By lowering the activation energy. By lowering the activation energy, whereby the activation energy is the barrier. You know, if you are to combine two things at once, you need the energy to make them one. So that energy which you must overcome before they become one is what we are calling an activation energy. So activation energy is the barrier. The barrier which reactants must overcome. Reactants must overcome to form product. To form products. So this is what we are calling that they act by lowering activation energy. They act by lowering activation energy. I can present it in a graph form. That if I have a graph like this, and we see on this graph, we have reactants. We have reactants A plus B, that before they form products, they must overcome an energy barrier. Before they form product A, AB. This is what, so if I have reactant AB, A plus B, before it forms AB, we need to overcome this barrier, which we are calling the activation energy. So from here up to here is the activation energy 1. Activation energy 1. This is without enzyme. Without enzyme. Without the enzyme. But if you bring the enzyme, in the presence of the enzyme, the energy or the barrier which these reactants must overcome, it is reduced. So you'll find you will need only a small energy to form products. Form products A and B. This one is in the presence of the enzyme. So from here to here, this is with the activation energy 2, which is the one with enzyme. So graphically we can see that at first without the enzyme, we needed much energy to move up to this side before we form product A and B. But when I introduced in an enzyme, we see that the enzyme has lowered the activation energy from this point up to this point. That's how they speed up the rate of biochemical reaction. That they have reduced the time we would waste moving from here up to here, and we have reduced it to moving only here. So that's why we say they act by lowering activation energy. That is the mechanism of action of the enzymes, how they speed up the rate of biochemical reactions. From here, there are terms we need to look at which are applied in enzymology. Terms applied in enzymology which we need to know as medical students. And these terms include, we are looking at the terms, definition of terms, whereby we shall need to know what is a substrate. We shall need to know what is a coenzyme. We need to know what is a cofactor. We need to define an apoenzyme. We need to define a holoenzyme. We need to define a prosthetic group. And lastly, we need to define what is catalytic activity. Catalytic activity of the enzyme. So these are the terms we should know because we apply them in enzymology. And a substrate is like a food for the enzyme. So a substrate is any substance that is acted upon by the enzyme. So substrate, in summary, we call it food for the enzyme, which the enzyme eats to form the product. Then the coenzyme, this coenzyme is the organic substance. It is an organic substance. And this organic substance is the one that combines with the enzyme to give it its complete catalytic activity. So this coenzyme is an organic substance that binds temporarily, or that binds loosely with the enzyme. It binds loosely with the enzyme. And example here, we normally have like NAD, nicotinamide adenine dinucleotide, acting as a coenzyme. Even we have PLP, which we call pyridoxyphosphate, acting as a coenzyme. So coenzymes, their role is to help the enzyme to do its role. That is a coenzyme. And the major thing, it is an organic substance, which is different from a cofactor. Because a cofactor is an inorganic. A cofactor is inorganic substance. And this inorganic substance, its role is to enhance the activity of the enzyme. It enhances the activity of the enzyme. And example here we have like magnesium ions, we have like iron being a cofactor, we have like manganese. All these inorganic substances, copper, they can act as cofactors. These are inorganic substances that enhance the activity of the enzyme. Then we have an apoenzyme. Apoenzyme, for it is the protein part of the enzyme. An apoenzyme is a protein part of the enzyme. Then a holoenzyme, for it is a catalytically active enzyme. Catalytically active enzyme. Or we can, in summary, we can say a holoenzyme is equal to apoenzyme plus a cofactor. If I get an apoenzyme, which is the protein part, then I add a cofactor, which is a non-protein part, then we form a complete catalytically active enzyme. These things are so sweet. Watch and see. So we are seeing a holoenzyme is a combination of an apoenzyme and a cofactor, whereby an apoenzyme is a protein part of the enzyme, then a cofactor is a non-protein part. So when you get a protein part, then you add a non-protein part, you get a catalytically active enzyme. Then another term is prosthetic group. As we saw that coenzymes, for them they bind loosely. Prosthetic groups, for them they are the organic substance that bind. These ones bind permanently. Bind permanently to the enzyme. So for them, once they bind, they never leave. That's why we call them prosthetic groups. For example, in hemoglobin, we know that at the center of heme there is always a prosthetic group called iron. And iron, it never leaves the hemoglobin. That's why we call it a prosthetic group. It binds tightly, or it binds permanently to the enzyme. That is what we call a prosthetic group. Then finally, it is the catalytic activity of the enzyme. Catalytic activity of the enzyme. Here we are measuring how many enzymes are required to catalyze 1 micromole of the substrate per minute. So the catalytic activity of the enzyme is the amount, how much enzymes, amount of the enzyme that are required to catalyze 1 micromole, to convert, can use the word to convert 1 micromole of a substrate per minute. Every minute, how many amounts of enzymes are required to catalyze 1 micromole of a substrate in every minute? That is what we call a catalytic activity of the enzyme. And this catalytic activity of the enzyme is measured in international units per liter. They are measured, these enzymes are measured in international units per liter. So the definition of a catalytic activity, it is the same as international units per liter. Whereby we are saying international unit per liter, international unit per liter. This is the units for the enzyme. They are measured in international units per liter, which is the amount of the enzyme required to catalyze 1 micromole of the substrate per minute. And they are expressed in international units per liter. So whenever you find an enzyme, their results are always expressed in international units per liter. So after seeing this definition of terms, now we look at classification. How are enzymes classified? Because we know enzymes are many in our body, there are many being used commercially, and how are they classified? So we want to look at the classification of enzymes. How are enzymes classified in our, and according to International Union of Biochemistry, we classify enzymes in 6 classes. That is according to International Union of Biochemistry, whereby this Union of Biochemistry came up with only 6 classes of enzymes. And these 6 classes of enzymes, they have a mnemonic. We can always remember them using a mnemonic O3. A mnemonic we use is O3. Whereby this O3, O stands for Oxidoreductases. O stands for Oxidoreductases. Then T stands for Transferases. Then H stands for Hydrolases. Hydrolases. L stands for Lyases. And another I stands, this I stands for Isomerases. Isomerases. Then this L stands for Ligases. So this is the mnemonic we can always use, OFIL, so that you can always remember all those 6 classes of enzymes under this mnemonic. So we want to look at, to explain one by one, and we see each class, what does it do? And we want to begin with oxidoreductases as number 1. Number 1, we have oxidoreductases. And these oxidoreductases, for them, they catalyze redox reactions. They catalyze redox reactions. What are redox reactions? Reactions containing both oxidation and reduction reactions. That's what they do. So they catalyze both oxidation and reduction reactions. For example, we have examples like glucose 6-phosphate dehydrogenase. Glucose 6-phosphate dehydrogenase is an example of an oxidoreductase, and another example we can have is lactate dehydrogenase. Lactate dehydrogenase. And these enzymes act in the presence of the coenzyme known as NAD, what we call the nicotinamide adenine dinucleotide. Whereby this lactate dehydrogenase, for it catalyzes the interconversion of pyruvate to lactic acid. And then glucose 6-phosphate dehydrogenase is the one that is catalyzing the reaction of the pentose or monophosphate pathway. So these are the 2 examples. At least under each class, you give at least 2 examples. And the examples we have seen is glucose 6-phosphate dehydrogenase and lactate dehydrogenase. And I have said they catalyze redox reaction. So what we call this reversible reaction. We can give an example of an equation that this example of the equation, I can put it here for visibility. Let me put it here. That if I have pyruvate, it is converted to lactate in the presence of lactate dehydrogenase enzyme, and this reaction includes a coenzyme, and this coenzyme here is NAD, whereby if we have NADH, then we form NAD+. This is what we call an oxidation-reduction reaction. Class number 2, we can look is at transferases. We want to look at the transferases. Transferases. And as its name says, trans means transportation, isn't it? So here we are going to transport functional groups. We are not transporting people, but we are transporting functional groups from one compound to the other. That is what transferases do. So transferases, they catalyze the transfer. You know, in enzymology, we use the word catalyze. They catalyze the transfer of functional groups. What are functional groups? Functional groups, we have like carbonyl groups, ketone groups, we have carboxylic groups. All those groups are the ones that are amine groups. Those are transported by these transferases. And they transfer these functional groups from one compound to another. Example, we can give an example of alanine aminotransferase. We have an example as alanine aminotransferase. We have aspartate aspartate aminotransferase. Transferase. This one is abbreviated as ALT, and this one is abbreviated as AST. These are the examples of transferases, whereby this alanine aminotransferase, for example, if I have ALT in the reaction, this ALT will catalyze the transfer of amino group from amino acid alanine. If I have an amino group alanine, it transfers it to 2-oxoglutarate, and we form pyruvate. We form pyruvate plus glutamate. How is it doing it? That this ALT is transferring the amine group. This alanine has the amine group. He transfers it from this amino acid and put it on 2-oxoglutarate. Then we end up forming pyruvate and glutamate. That is the role of transferases. The third class is Lyases. The third class is Lyases, and the Lyases, I can put them here. It is Lyases, then put it here for visibility, so that you can see them very well. Lyases is number 3, whereby, as the name, this name comes from the word lysis. I'm seeing this name comes from the word lysis, whereby lysis means breaking down. So what Lyases do, they break down compounds. For example, these Lyases, for them, they catalyze the breakdown of bonds between compounds. And these are always bonds between carbon, oxygen, carbon, it can be carbon, nitrogen. So you find these Lyases, they come and they break the bonds non-hydrolytically. They start breaking the bonds. Then they lead to separation of these compounds. And examples of Lyases, we have example like Hydrolase in glycolysis. Hydrolase is an example of Lyase. And we have also agenosuccinate in urea cycle. Agenosuccinate, Lyase. Agenosuccinate, Lyase. What does Hydrolase do in Krebs cycle? When we have fructose 1,6-bisphosphate, which is a 6-carbon compound, we see it Hydrolase breaking it down, cleaving it into 2. And it is cleaved to form dihydroxyacetone and glycerolaldehyde 3-phosphate. So that's why we are saying they break down bonds between compounds. And I told you in each class, at least endeavor to give always 2 examples, at least in an exam. Class number 4, we can look at hydrolases. Hydrolases, and you know, it has the word hydro. And this hydro means what? Water, isn't it? So these enzyme, class of enzyme, they catalyze hydrolysis reactions. They catalyze hydrolysis reactions. What are the hydrolysis reactions? Meaning they involve addition of water. These enzymes, they break the bonds by addition of water. By addition of water. Hydrolases, for them, they break the bonds by addition of water, but Lyases, for them, they break the bonds non-hydrolytically. Non-hydrolytically. They don't involve addition of water. And sometimes the end result of this product of Lyases is always double bond. Never forget that when Lyases break compounds non-hydrolytically, they lead to formation of a double bond. Then for hydrolases, for them, they break the bonds by addition of water. And here examples, we include all digestive enzymes, enzymes like pepsin, enzyme like amylase, enzyme like sucrase, and so on, maltase, lactase, all those are hydrolyses, they are hydrolases. Then another one is urea. Then another example I can use of a hydrolase is ALP, alkaline alkaline phosphatase. Alkaline phosphatase is also a hydrolase. So these are the examples. So whenever you ask examples, or in an exam they ask you that the following are hydrolases, except you should be knowing the examples. All digestive enzymes in the GIT, the hydrolases, then including urea and alkaline phosphatase are all hydrolases, whereby they break bonds by addition of water into the bond. Then in class number 5, we have isomerases. And these isomerases, for them, they catalyze intramolecular arrangement. They work on what we call intramolecular arrangement. Intramolecular arrangement of molecules within a compound. Of molecules within a compound. What am I meaning? That if I have a compound ABC, this ABC catalyzes the arrangement of these molecules from different positions. For example, A is in position 1, B is in position 2, C is in position 3. In presence of an isomerase, we see the B which was in position 2 going to position 3, and the C which was in position 3 going to position 2. Intramolecular arrangement. Then I form a product A, C, B. That is what we call isomerases. And examples of isomerases include triose phosphate isomerase, triose phosphate isomerase. This one is in Krebs cycle. We have aconitase in this triose phosphate isomerase is in glycolysis. Then we have aconitase in Krebs cycle. We have even phosphofructomutase in glycolysis. Those are the examples of isomerases. Lastly, let us talk about ligases, which is the 6th class of enzymes. Ligases, and these ligases for them, they catalyze the joining of bonds. Ligase is something that can join two things together to form one compound. So what ligases do? For them, they catalyze the joining. They catalyze the joining of two compounds together to form one compound. So when I have two compounds, for example, A plus B, these are two compounds. In the presence of a ligase, they are joined to form AB. But this reaction of ligases, it involves hydrolysis of ATP. If I have a ligase here, you need a hydrolysis of ATP because we need energy. As I told you in the beginning that to join two compounds, you need energy. And this energy is provided by ATP, and we form ADP plus phosphate inorganic to form our one compound. We had two compounds A and B, And the final result, we are forming one compound AB in the presence of a ligase, which involves hydrolysis of ATP, or breakdown of ATP. An example of these enzymes, we have like pyruvate carboxylase, enzyme. We have glutamate synthetase, glutamine synthetase as examples of ligases, and even not forgetting the ligase enzyme in DNA, which joins the two strands of DNA together. That is the ligase enzyme. It is also part of the ligases. So that is what we had in introduction to enzymology, where we have talked about what an enzyme is, which we said it is a biological catalyst. We have seen the properties of enzymes. We looked at different terms which we use in the enzymes. And we have also seen, we even saw the mechanism that these enzymes act by lowering activation energy, which is activation energy is the energy barrier that the reactants must overcome to form products. Then we have gone ahead to look at the classes, 6 classes of enzyme according to International Union of Biochemistry, where we use the mnemonic O3, whereby O stands for Oxidoreductases, T for Transferases, H for Hydrolases, L for Ligases, I for Isomerases, and another arrow for Ligases. Thanks for listening. Continue to subscribe. Don't forget to subscribe and put your comment and like the video and share to all. Muhumuza Naphtali, sciences.com at your service. See you next time.